Welcome back, students, to the current topic, the water vascular system in Echinoderms. In this topic, we will just discuss about the water vascular system in Asteroidea. The water vascular system is also called as an ambulacral system, and this is a unique feature of Echinoderm. And this type of system, it is absent in any other animal group. It is a system of canals and associated structures filled with a watery fluid containing corpuscles. And this entire system is formed from the hydrocele, which is comprised of canals, and it has a lining of ciliated epithelium. The water vascular system comprises of mainly the 5 different parts: madreporite, stone canal, ring canal, radial canal, and tube fate. These are the basic constituents of water vascular system, and apart from this, there are the certain other components also which adds to this water vascular system. So just see in this diagram, this is a picture of starfish. On the aboral side, you can see a small spot, and this spot, it is a madreporite. It is found on the aboral surface, and on the oral surface you can see the certain structures called as a tube feet, and these are the two structures which we can see on outside of the organism, which is a part of the water vascular system. Rest all other parts are arranged internally. This diagram shows the magnified view of madreporite, this portion, and here you can see the certain ridges and pores, and which arises as a plate-like structure on the aboral side. This is a view of madreporite. In this slide you can see a sectional view of water vascular system. Here it is a magnified view of the madreporite. This madreporite arises, or we can say that the madreporite is seen interradially between the arms on the disc region. This is a central disc region, and these are the arms of starfish, and in between the arms interradially you can see the at the base, you can see the base of the arms, you can see the madreporite. The water enters to this madreporite, and madreporite is also called as a sieve plate, which acts as a sieve and which drains the water into the canal system. And from the madreporite it reaches a canal system. This is the umbilical canal system, and it ends in tube feet. You will just see in detail the water vascular system in starfish. The water vascular system, as said earlier, in starfish mainly comprises of following parts: the madreporite, the stone canal, the ring canal, the radial canal, the lateral canal, and ends in tube feet. There are the certain additional structures called as Clipman bodies and Pauline vesicles. This madreporite is a specialized structure, and it is a magnified image of madreporite. As you can see, the numerous pores and grooves, or in turn we can see the madreporite is formed of ridges and grooves. It's otherwise also called as a sieve plate. This diagram shows the vertical section of madreporite. On this madreporite, these are the pores. These openings are the pores of madreporite, and following the pores you can see small canals which arises from these pores, and these canals are called as a pore canals, and these pore canals unites together to form the collecting canal, and this collecting canals opens into the ampulla region of the madreporite, which is followed by the stone canal. This is a part of the stone canal, and this stone canal, as you can see here, it is a S-shaped canal extending from the madreporite. The wall of the stone canal, it is strengthened by a series of calcareous springs and hence the name. And in this image you can see the lumen of the stone canal is completely or incompletely divided by long ridges. These are the ridges and lamina. The lumen, it is divided into ridges and lamina. This stone canal opens to the ring canal. This ring canal, it is a wide and somewhat pentagonal in shape. It is lying around the esophagus. In the center you can see the esophagus, and surrounding the esophagus there is a ring canal. The 5 corners of the pentagonal occupies the radial position and it receives a stone canal interradially. Interradially, it receives a stone canal. This is a radial canal, and interradially, in between these two radial canals, the stone canal enters into the ring canal. Here you can see the 5 radial canal. This 1, 2, 3, 4 and 5. 5 radial canals are there in Asteriasis. And this radial canal, this runs forwards. It runs forwards in 5 angles of the ring canal externally. Each radial canal runs into an arm above the ambulacral groove and terminates in a short tentacle at a tip. This is a short tip, that is, it ends in the tube feet. This radial canal opens into the small, short lateral canal and it enters into the tube feet. This tube feet comprises of the 3 regions, mainly the ampulla, podium and the sucker. So the lateral canal of each arms or each radial canal enters into the lateral sides 2 short lateral canals. These are the, as you can see there, these are the short long lateral canals which is alternative in arrangement. You can see alternative, one after the another alternative you can see, and each lateral canal carries a tube feet at its tip. The lateral canal are provided with the valves which prevents the flow of water back to the radial canal. So once again, the lateral canal are provided. These are the lateral canal, also called as a podial canal. These are supplied with or provided with the valves which prevents the flow of water back to the radial canal. The valves are placed at the junction of the lateral canal and the ampulla. Here in this portion you can see the valve at the junction of the ampulla and the lateral canal. So apart from these structures, on the ring canal you can see the Tickman bodies. These are the Tickman bodies, racemose-shaped Tickman bodies, and also the Pauline vesicle. These are the Pauline vesicle. These Pauline vesicles, these are the flask-shaped structures, contractile structures, which helps to store the seawater and also to help the hydrostatic pressure inside the system by quickly emptying the water. But some authors say that they help in the lymphatic status, producing the amoebocytes. Anyway, this bag-shaped structures, Pauline vesicles, are seen alternative with the radial canal. You can see this alternating with the radial canal. Then there are the Tickman bodies, are yellow in color, glandular bodies, which are seen on the ring canal. This varies depending upon the different echinoderms. These structures are believed to be the lymphatic glands involved in the production of the copper cells found in the water vascular system. So pigment bodies and Phylum vesicles are their functions to perform in the water vascular system. So actually this water vascular system helps in the locomotion of this echinoderm. This when the animal moves by the 2 feet. It is said that the animal moves with the help of this 2 feet. The water which enters through the madreporite passes through the stone canal. Then it reaches the ring canal. From the ring canal, it reaches the radial canal. From the radial canal, it reaches the podial canal or the lateral canal, and finally into the ampullae of the podia. As the ampulla contracts, the water is forced into the 2 feet, and this 2 feet which gets elongated. So it gets a suctional force. The sucker-like tip of the 2 feet touches the substratum. This end of the 2 feet that touches the substratum and contracts again, and the fluid goes back into the ampulla and the body is drawn forward, bringing about the locomotion of the starfish in seawater. So actually the main function of this water vascular system is locomotion. It is how the organism moves, and this is a flow of vascular system. Flow of water in the vascular system is depicted, as you can see, from the sea plate to the ring canal, from ring canal to radial canal to the ampulla, and finally to the 2 feet.